With Doubtnut, get instant video solutions to all your maths, physics, chemistry, and biology doubts. Just click the image of the question, crop the question, and get instant video solution. Download Doubtnut app today.
Hello students, the question here says that sequencing the whole set of genome that contained all the coding and the non-coding sequence and later assigning sequence with functions are referred to as which of the following? While we see when we talk about HGP, it was a human genome project in which the sequencing of the DNA bases was done. While we know in humans we have 6.6 into 10 raised to power 9 base pair in a diploid cell content, while this is 3.3 into 10 raised to power 9 in case of a haploid. So while we need to work it out, we found that sequencing from a single cell required $9 billion because every base pair required approximately $3. So this was considered to be a mega project here, while there were two approaches to this project. One was simply sequencing the entire set of DNA, whether it is a coding sequence or the non-coding sequence. While we know that only 2% of the DNA is the one which codes for proteins, rest of it is the junk DNA, which has functions such as the evolution and other functions, and these sequences are called as the junk DNA, right? So here sequencing the whole set of DNA, whether it is coding for proteins or not coding for the protein sequences, this method was called as sequence annotation. So in sequence annotation, we are going to assign the different regions with functions also. So here we see option number 1 here becomes the right answer for this question, while the other method in the sequencing of DNA is the express sequence tags. That is the ESTs. So express sequence tags means that only the coding segments of the DNA will be sequenced. So this is express sequence tags or ESTs, right? So whole genome in this case was not done, only the coding part of the DNA was sequenced in this. So this is just opposite to what was done in the sequence annotation. So this is the right option, whereas PCR is polymerase chain reaction, which is to make multiple copies of the gene of interest used in the biotechnological processes, whereas northern blot is a test which is used in the AIDS, and microarray is also the wrong option. So this is not related to the HGP. So these three options are wrong, while the only right option here is option number 1. This is all about this query. Thank you.
For class 6 to 12, IIT JEE and NEET level. Trusted by more than 5 crore students. Download Doubtnut app today.